Okay, good morning. Let's have a class. And first of all, I just remind you, on the eCampus, I have been putting some of the after-class reading material. Please go ahead and read, because these are basically is my class lecture. I coming from different textbook, and I combined it. So it's a very good after-class reading. And also this case study, we're going to talk about something later, but you can go ahead and study it. Okay? I have set up with one fact sheets, the second fact sheets, and also have a third fact sheets. This is Corynebacterial diphtheria, we already talked about. Klebsiella and a case study, exam review. Some of them will be directly in the exam. So it's just want to let you know, I want you to study. Okay, it's very important. So today we're going to talk mainly about Clostridium. But before that, we will go over something else. So the title will be Human Disease Caused by Bacteria, second section. Very obvious, human digestive tract. And everybody knows you have oral cavity. There's lots of the bacteria going to grow there. We already said it is we call Viridime streptococcus. Lots, and we will do isolation some of them in the lab section. Esophagus, that's where your food goes here. So you go liver, stomach, pancreas, large intestinal, small intestinal, appendix. We don't care about most of the part. This is not a anatomy class. We care about the bacteria. So, very simple idea. Stomach, very few bacteria. Okay? How few it is? 1 to 100 cells. Why? It's very simple. Stomach, acid, pH 1 to 2. This is extremely low acid. It's a first defense system. We can say it's a physical defense system for the bacteria going to the intestine. Stomach acid could kill lots of the bacteria. So in other words, lots of the food pathogen, foodborne pathogen, why they will cause a problem? So you have to combat the low acid environment. So we can say more or less they are a little bit acid resistant. Small intestinal, there are not many, but it's already being much higher than stomach. It's 10 to the power of 4 cells. When you look at large intestinal, this is huge. That could be up to 10 to the power of 12 or 14 cells per gram. This one is a major research topic these days, is intestinal bacteria flora genome. And people think your whole body, the function is coming from there. It is a normal, balanced microflora community in the body. It's a center of everything. Even as important or more important than your heart, than your brain. So lots of the research has been going on. Okay, just want to let you know. You can do whole genome sequencing. In the older time, we do a DGGE methods or PFGE methods. The DNA fingerprinting, right now we can directly do the whole genome sequencing. It's lots of the topic, could be covered in some other classes. But here we want to mention, majority of the large intestinal bacteria there is anaerobic. This is key. 67% is anaerobic. The left around 30% is aerobic. And our fecal sample, or you poop, dominant with all the bacteria. So you know in our health science center, lots of the research what we could do, use human subject. Let's say we let them orally take a lactobacillus, or like a fermented products, and we collected the fecal samples from different family. We can do the analysis. Okay, that's a lot of things to do. So this slides, it's simple, tells you normal flora in our alimentary tract, but lots of the science behind that. Okay, just want to let you know. And our large intestinal is a huge community of microbacteria, of microorganisms, that is the center of our body. Okay, next one. These slides will tell you one very important concept, which is called food intoxication. And I briefly mentioned, I want to talk a little bit more. This is called food intoxication versus food infection. So first of all, we will talk later in the exam 4 about the foodborne pathogen. Let's say you have E. coli 0157H7, Salmonella, Listeria. These we call food infection. What this means, you are eating bacteria. So it seems like if it's contaminated with beef, with poultry, chicken, with hot dog, you eat it, you got sick, you're eating the bacteria. So how we solve the problem? Thoroughly cooking. So you thoroughly cooking, you will destroy them. That's called food infection. Now, food intoxication, the example is right here. You have Staphylococcus aureus, you have Bacillus, and you have Clostridium. That will be a major topic today. These, if it's contaminated in your body, you are eating toxin. So what this means, the toxin is already there. Lots of them are heat resistant. So even you cook it, you may not can solve the problem. Another thing is that the onset time is very short. Somehow 1 to 6 hours, you got vomiting, you got diarrhea, and you got nausea. So that's something I want to say, the difference is there. The difference is here, okay? And another thing I also want to say, very ironically, if you get contaminated with Staphylococcus aureus, with Bacillus, you don't need to report to CDC. When you contaminated with E. coli 1577, Salmonella, Listeria, the doctor has to report to the CDC. Okay, so something is really different. But the bottom line here is that food infection, eating bacteria, thoroughly cooking can solve the problem. Food intoxication, you're eating toxin. Sometimes the toxin is heat resistant. Okay, so the prevention, the other prevention, we said the process preventive control is important. You cannot rely on everything finally is doing the cooking. And sometimes even a cooking will cause more problem, and we will be mention that. Now this is a very good slide to talk about these examples of these food intoxication pathogen. So we learned something already. Bacillus cereus have window spore, Staphylococcus aureus we already mentioned, and the Clostridium botulinum we're going to talk real quick. But basically, lots of these have the symptoms. It's like vomiting, diarrhea, nausea, abdominal pain. These are very common symptoms for foodborne pathogen if they contaminated in your body. Okay. Today we want to make sure we talk lots of things is about Clostridium. Okay, we want to go here first. So I want to completely remove this. Then we want to spend time talk Clostridium. It's a major We can say a foodborne and a human pathogen. So Clostridium. I just write Clostridium SPP. First of all, Clostridium is gram-positive, large shape, little curved. This pathogen is very different from others. It is an endospore forming bacteria. So remember the endospore. Why endospore can survive in the harsh environments? Calcium diproconic acids and very small acid-soluble DNA protein. We already mentioned it. We will talk about it in the lab again, so review again, so we briefly go over it. And this is possibly subterminal lots of the time, the location. It's an endospore forming bacteria. The bacteria what we talked before, Streptococcus, Staphylococcus, Haemophilus influenza, Neisseria, those are not endospore forming. This is an endospore forming bacteria. Most of them coming from soil. Okay, that's the key. Number 3, they are all strict anaerobic bacteria. No oxygen. Remember in the exam 1, I have an extra point. I asked why Clostridium cannot survive in the presence of oxygen. Lots of you didn't do very well. They say because Clostridium is an anaerobic bacteria. And I often make a joke, this is like ask you why it's an apple because you say it's not an orange. But you need to tell me why. So we learned it in the class already. Why it is a strict anaerobic bacteria? Because aerobic bacteria, they need SOD and catalyst, is that right? To destroy or hydrolyze the toxication material. This typically is superoxygen, this typically is hydrogen peroxide. Those are harmful material for the bacteria. And these enzymes have to hydrolyze them. Then they can survive with the presence of oxygen. And without these enzymes, they cannot survive in the presence of oxygen. And oxygen for them is the toxication. So the answer is no SOD, no catalyst. That's why I said it's a very short answer. Okay? So this is an overview for Clostridium species. And we would love to talk about these one by one. So let's talk about the first. Let's talk about this one first. Clostridium difficile. Clostridium difficile, you can say the acronym is CADIF. Just say CADIF. It's simple and easy. So what is this? What is this one? Where does this one cause a problem? This is a nosocomial infection in unsanitized hospital facility, including nursing home, they contaminated with people is extensively using antibiotics. For example, amoxicillin ampicillin for 4 to 7 days, sometimes more than that. So very simple. A 70-year-old grandma in the hospital, in the nursing home, have pneumonia. Then the doctor using antibiotics, using amoxicillin or penicillin very intensively for 7 days. She will have water diarrhea. And then we have to do some tests. What we do the test? We do sigmoid scopic exam. We find it is a pseudo membrane colitis. So if we go to a colon area, we do a colonoscopic exam. What they will find is in the colon area started bleeding. And you will see some smear dots like that. It's a swelling, it's a dead white blood cell, looks like that. It's a pseudo-membrane colitis. Now, this is a symptom. So how are we going to do that? The stuff, the toxication found from Cadif, it is also an AB toxin. Same as Corynebacterial diphtheria. This is toxin domain, cause inflammation, and this is exactly what happens. And the B toxin is transmembrane domain and will cause a bleeding. So how are we going to do the test? AB toxin test. If you are doing the send the stool sample to the reference lab or even using a rapid testing kit, you find it have an AB toxin and accompanied with the pseudomembrane colitis. This lady is diagnosed as Cadif. It's very severe and interesting results. I want to say this one. The Cadif does happen in the nosocomial, which means in the hospital. Sometimes in the nursing homes, because lots of people, they fail to do the sanitation standard operation procedure, and even some of the operational facilities, if they didn't do very well, they could be contaminated. So that's caused a problem. Now, we want to talk about something is recently. Cadif is not only happened in the human being, it's also going to be transferred from the fecal sample or the stool sample from the human being to the food supply chain. So Cadif, in the recent 20 years, we also call it a new emerging foodborne pathogen. So which means this guy could be not only in the hospital, it's also in the food supply chain. I remember the Department of Agriculture, National Institute and the Food and Agriculture Foundation, which supplies the research funding for all the land-grant universities in the United States, they funded a couple projects in 2012, which is regarding the extension and the research integrated activities to prevent the Cadif in the food supply chain. Because it's very easy to transfer through the fecal contamination. Okay? So this is the important thing. Now, we wanted to say something about treatment. If somebody has Cadif, what we should do the treatment? It depends how severe it is. If it's very light symptoms, we could do drinking fermented, for example, lactic acid bacteria liquid or beverages. Why? You intensively using amoxicillin, ampicillin, basically the normal bacteria in the intestinal is already been dead. So the normal flora is affected. They are gone. I put a beneficial bacteria, lactic acid bacteria there, they will help. So that's the easiest way to do it. The second one, it's an ironic one. Using another antimicrobials, vancomycin. So what happened is that you said, I'm not using amoxicillin and ampicillin. I'm going to choose another antibiotics. I use vancomycin. But it sounds very ironic. Why? You already said this is intensively using antibiotics for 7-10 days. How come you're going to use another antibiotics? The reason is the antibiotic mechanism of vancomycin and penicillin are different. This is basically is beta-lactam, and this is attacking the peptide cross-linkage of the modified sugar of peptidoglycan. And we will talk about next week. So the second method is using another antibiotics, which is vancomycin. But if it is a very severe infection, what we have to do? Fecal transplant. This is horrible. Think about all your fecal sample going to take it out. Other people's poop going to put on your body. It seems horrible. That's called a fecal transplant. But we have to do it because the idea is to put normal bacteria from intestinal go back to your body. Okay, so these are what we have for the Clostridium difficile. It is a hospital-acquired contamination. Okay, major symptom, pseudomembrane colitis. Okay, the second one. We're going to go back to the earlier. Clostridium botulism. This is the first one. Second one, botulism. Okay, for Clostridium botulism, we want to talk about three things. There are basically three categories of botulism. It is wound botulism, infant botulism, and food botulism. So, what is a wound botulism? You have cuts, wounds, bites on your skin surface. Let's say your skin surface have this cut, a very thin. This will be a minimal oxygen environment. What this means, anaerobic bacteria will dominate. Okay, Clostridium botulism will be there. If this would touch with some of the soil, decayed metal tools, you will have a problem, especially for drug abuser. If a chronic drug abuser, this will be a high risk group for wound botulism. Second one is infant botulism. How you understand that? It's a very simple sentence to describe we will say you never feeding less than 1 year old baby honey. Otherwise, you have floppy baby. So this is the sentence, a joke, but it's well described. Clostridium botulism, which is infant botulism. So why is when less than 1 year old baby? Less than 1 year old, your normal bacteria in the intestinal has not been established. So you get a little bit from the mom, but you have not been established very well your own intestinal micro community. Honey is coming from soil, the original source. It's very easy to contaminate it, and it's a high sugar amount. It's a good nutrition for the Clostridium, the endospore to dominate. If you do that, you will have a very floppy muscle. Why? For the muscle contraction, if we do this contraction, what is the transmitter, the neurotransmitter? Acetylcholine. And this Clostridium botulism toxin will attack it. So the muscle cannot contract it. The muscle is completely relaxing. This comes off with floppy muscle. That's what the picture looks like. Okay? So that's called infant botulism. But I'm a food microbiologist. I'm really interested about this last one, is food botulism. What is that? This relates to one group of the food. It is very popular in West Virginia area, canned foods. So first of all, canned food, it's fit to one category. We call it is low acid food. What this means, pH below 4.6, more than 4.6, water activity less than 0.85. This is a perfect environment to favor endospore bacteria Clostridium botulinum to grow. It's very, very interesting. So, there is some of the misunderstandings they do the processing. So for example, I'm preparing a canned tomato. Okay, I put a tomato there. The tomato piece there. Where the tomato comes from? The tomato comes from the soil. Even you wash it, you sanitize it, more or less there's a bacteria there. Okay, then you put it there. Lots of people not even really wash. They do blanching 2 hours, just boil it, this, and then followed by cold pack. It will fail. Because even you do boring, the endospore will not be killed, and then you immerse into the ice pack, the cold pack. It's not going to work. So what we have to do? We have to use pressure cooker. The pressure cooker, 121 degrees Celsius, 15 minutes, and 15 psi. That is the bottom line requirements. It's a sterilization process which is killed everything. Now, how do you make sure the pH is below 4.6? You need to add lemon juice or concentrate or vinegar to decrease the pH less than 4.6. Now at West Virginia area, the West Virginia Department of Agriculture do have a regulation for canned food. What is the standards? Require you pH less than 4.0. It's a protective line because it's a little bit strict than 4.6. This is a way we could to prevent the botulism. However, the canned food, it is very tricky lots of the time. Okay, you see the picture there, how tricky it is. When you ever go to a grocery store, they usually have some markers will tell you the canned food you cannot eat. Number one, if you see the liquid is very milky, which means it's contaminated, you already see the extract, which means the liquid is pouring here, you cannot use it. And you see this lid, which is dented, you should not use it. Okay? Even the edge, you see it's concave or dented, you should not use it. So these are the basic information for Clostridium botulism. Now, Clostridium botulism, they do have a toxin. The toxin is A, B, C, D, E, F, G. There are so many toxins. But in the United States, the most important is A, B, and E. The B toxin typically responsible for 40% of the case in the US. And the E toxin is very typical for canned fish product. Now, how are we going to do the treatment? If you have a botulism contamination, you go to see a doctor, you need to use antibiotics. Now, obviously, it's a bacteria, but because it has a toxin, you have to use antitoxin. Now, where the antitoxin comes from? The hospital has to go to CDC. In the southern part, the CDC is located in Atlanta, Georgia. They have to quarantine the airport. Then the airport have to shipping immediately anti-equine toxin. That's a toxin originated from horse. Go to the hospital, then you're going to have an injection of antitoxin immediately at the same time with antibiotics. It is very important that you have to do both because this is a toxin have to prevent all of the muscle contractions, the issue. Now, I also want to mention Clostridium botulism. This thing is important. This thing is severe. Why? This is not like what we have, just a nausea, diarrhea, or this. This will cause neurological symptoms. What are the neurological symptoms we'll have? Typically, difficult speak, difficult swallowing, and double vision. That's why it's severely, you have to be paying much attention to that. So the picture here, which is showing you the floppy baby, the infant botulism, Wood's botulism coming from cuts, bites, especially for chronic drug abuser, and canned food and a high pressure cooker, This is for the food botulism, especially for the canned food, and the standards here in locally in West Virginia is the pH has to lower than 4.0. Okay. Next one, Clostridium tetanus. So I'm gonna remove this guy here. Clostridium tetanus is not that difficult to understand. I believe we mentioned and you heard about that before. Tetanus. This will cause a severe symptom starting from wounds. Very similar to the wound botulism here. It is coming from the wounds immediately, and the toxin is very invasive, could be transferred, and the very typical symptom is lockjaw. Now, why? Tetanus toxin is existing. And what happened is, the muscle, they will need something called glycine. The glycine will cause muscle relaxing. The tetanus toxin is block the glycine. So what happened? The muscle cannot relax, can only do contract. The contract will cause lockjaw, and severely will cause stiffness of your respiratory tract muscle, which is peripheral muscle. So finally, you cannot even breathe. That's why it will cause people to die. That's a very simple principle behind it. Now, Clostridium tetanus, we already mentioned. If you get contaminated with tetanus, it's very difficult to do. It's very difficult to do the treatment because the toxin is translocated too quickly. So we have to do the prevention. Luckily, we have one. We mentioned about that one. This is a toxoid chemical-like vaccine. We need to do a booster. We mentioned because it's only activate T cell. But luckily we have it. Diphtheria. What is T? Clostridium tetanus. This is important. That's why DTaP vaccine is very important in the immune scheduling because it covers those three things. This is pertussis. Is that right? So that's what we have for the tetanus. And I have a very nice picture right here to explain to you the principle behind botulism and the tetanus. Look at the botulism, what it looks like. You all have a central neural system. The motor neuron and the transmitter. There is basically two important neurotransmitter, which is acetylcholine and glycine. Acetylcholine signals muscle to contract, and the glycine is for relax. Botulism is block releasing acetylcholine. So you cannot do contraction. You're always relaxed like this because acetylcholine is blocked. And the tetanus in the opposite direction, they block glycine. So you cannot do relaxing. You can only do contracting. You can only keep like this. Then if it's infected your muscle, the breast muscle, the whole body cause is lockjaw. Okay? So you can see botulism block release acetylcholine and tetanus toxin blocks release of glycine. Okay? So muscle M contracts, muscle O cannot relax. So this is a very good picture to tell you the principles and behind botulism and tetanus. Okay, last one. This is another important we want to talk about is Clostridium perfringens. Clostridium perfringens. Clostridium perfringens will cause a necrosis decay. We call gas gangrene. G-R-E-N-E, gas gangrene. So, first of all, I want to say, although Clostridium difficile botulism, you're going to testing the toxin. Rapid testing is toxic. Clostridium perfringens, you have a very easy, quick methods. Because on the blood agar, if you do a streak plating, what will happen? You will see it is connected transparent zone. So on blood agar, it is double beta hemolytic zone. So it is very easy to do incubation and get recognized. Now obviously this blood agar you have to put in an anaerobic jar, okay, or anaerobic chamber, and we mentioned need to take out all the oxygen and put methylene blue as a gas indicator, because methylene blue is colorless, which means the oxygen is gone. So you have to put it there. Double beta hemolytic zone. Now why it will cause gas gangrene? This is what happens is, if this is a muscle, I have one cuts or when wood comes in. In this area, this is a low oxygen. So the bacteria will start to survive. Once the bacteria start to survive, they will generate the enzyme. The enzyme will separate all these muscle pieces. All these muscle pieces will separate. And then they will generate a gas surrounded here. When you see here, it will be the gas bubble generator. The gas bubble generator. Here is the muscle. The major components of the muscle is glucose, is sugar glucose. In this low environment, what they're going to generate is kind of like a fermentation. The fermentation will generate acid and will generate a gas. So the gas could be separated in this muscle, and they generate a pressure. Now, if they generated a pressure, the same as we talked about before, if you have a blood stream right here, and this is your liver, and you have the pressure goes, bottom goes down, certainly the pressure going to the liver is not enough, which means you do not have enough blood supply to the liver. The liver will cause decay, and then what happens? You have a shock. It's very simple toxication shock. That's gas gangrene. That's the severe symptoms of the gas gangrene. And this is told you only the necrosis is on the surface. But it's going to be more severe than that. Now, where the gas gangrene comes from? This is something very interesting. There's a true story. It's not my colleague at Western Kentucky University. That's my second job before I came here. I went there 2 years, teach microbiology. My friend like Mark Carlson told me there's a story. A faculty, his friend at University of Louisville, not far from here, They are a study for the social science. So one day in the morning, they are using a hot pot. They are preparing a hot pot. In the hot pot, the guy put something like beef stew. And they put carrots. They put carrots there. They put tomatoes. Of course, they put water there. And they do is a long time, low temperature cooking. Because they set up only 50 degrees Celsius. And the whole family is go to hiking. He and his wife, and with a daughter and with a dog, they go hiking. After a couple of hours, 6 hours later, they're hiking when they go home, and they start to eat the soup. Everybody have a one bowl. 2 hours later, they all have a vomiting diarrhea. They go to emergency room at University of Louisville Medical Center. And what happened is Clostridium perfringens contamination. It's just this simple example could cause a problem. Why? You have a beef stew there. Beef stew, you cook it. You add a liquid. It will generate beef extract. That is why our bacterial media is very similar to a beef soup. It's brownish yellow color. It's a high nutrition ingredient to bacterial growth. You have carrots, you have tomato. They all coming from soil. The soil is a major reservoir for Clostridium, botulism, perfringens, and everything. And the key thing is here, you do low temperature, 50 degrees Celsius, and a long time. This temperature is enough to kill lots of the vegetative cells. And this is also a stress environment for endospore bacteria, so they don't have a competitor. All the normal bacteria are gone. And in the stress environment, they start to form in the endospore because of germination, sporulation, and then it starts to grow endospore. And more are going to be dominant. So later on 6 hours, this soup is filled with bacteria endospore. Because a long time is a cultivation. You have enough nutrition, you have this low temperature killing all the competitive vegetative cells. That's why it's dominant. And it will cause a problem. So that's why I told you, the Swiss vide cooking, use low temperature, long time cooking, you absolutely is not do that often. Because that's lower temperature, long time cooking. It's a perfect environment for endospore to forming. So what we should do, and very simple, avoid dangerous temperature zone. And what is that? You may heard, but you never recognize. It's 40 to 140 degrees Fahrenheit. 4 degrees Celsius to 60 degrees Celsius, it's a dangerous temperature zone. So you need to keep cold food in the cold environment. 0 to 4 degrees Celsius, below it. You need to heat food products is enough heat, more than 60 degrees Celsius. But of course, you cannot do frozen. Is that right? Frozen will generate a crystal. So crystal, it's a sluggish of the ice crystal will damage the tissue of the food products. Now, obviously, you cannot cook in too high temperature because we talk about tenderness, flavor, juiciness. The tenderness of the beef products will be damaged. So that's why there are lots of the research going on there to talk about. People think about antimicrobials, use a high pressure cooking, use a different engineered methods could solve the problem, but still they failed, and the dangerous zone we have to avoid. It's very easy to remember. It's 40 to 140 degrees Fahrenheit and transfer to 4 to 60 degrees Celsius. Okay, it's a very interesting topic we talk about that. So that is Clostridium. So we finished talking about these Clostridium. We want to go over something else here. This table 28.5 is really another topic. This is talk about the foodborne pathogen is food microbiology. We have a Dr. Jacek Jachansky have another class which is talk about all these things in more detail. It is actually my research topic, so we just go really simple here. Salmonella typhimurium, Salmonella typhi. We want to make sure you understand when we talk about Salmonella, we are talking about Salmonella is Salmonella non-typhi. If we want to talk about Salmonella typhi, that's a different story. That is related to a very magical woman called the Typhi Marrow. So what happened is, she is a cooker from New York City. So the first is her job is doing the cooking in the restaurant. And herself is okay, only mild symptoms. Nausea, vomiting, but the people surrounding her are going to die. Then she was quarantined, has to do quarantine. And then, after a couple of years, she recovered, then they go to work in a bookkeeper, in a library. And what happened is the people surrounding her, a couple of people also infected and died. So finally, the NYPD have to strictly quarantine her to Long Island somewhere in the suburb. And in 1960s, she died, she passed away because of the liver disease. And then people started to do the research. And until very recently, the Stanford University, the researchers find there is a Salmonella typhi, which is hidden in her spleen, which is packaged with a macrophage. And because of that, they come out with a very special species called the Salmonella typhi. But we are talking about Salmonella typhimurium, Newport, Thompson. It's affected mainly is a poultry, egg, or meat products. It is Salmonella non-typhi. So I just want to let you know it's a different story between Salmonella typhimurium and Salmonella typhi. Okay. Vibrio chlorium, it's most likely is a seafood coming from the jellyfish. In the deep ocean marine area, they will be very easy to be contaminated. If you have Vibrio chlorium, you will lose tons of water in a couple of minutes. So, Vibrio, if you got that problem, is not using antibiotics. The first thing you need to do is rehydration, which means put water back. We have a slide to talk about that. Okay, the next one, Shigella. Shigella, this is the one, very similar to E. coli. It will generate Shigella toxin. But Shigella is very typical for daycare kids. So in the daycare, then they're 3 years old, they go to poop. And then they just do whatever, then stealing, then go to touch everywhere, then everybody gets contaminated. So Shigella basically is a high-risk group is daycare kids. Campylobacter, we will talk in detail. It's a microaerophilic bacteria. It's a poultry is a big topic. E. coli we already said. We're going to talk later. Okay, Yersinia. Yersinia, it's talk about the milk, but not really. Yersinia is for pig sheetlings, which means pig intestine. You never eat it. I usually eat it. If you go to Pittsburgh, The Chinese restaurant at Forbes Avenue or surrounding the University of Pittsburgh, you could get a bowl of a very nice noodle soup with pig chitlins. And you never eat it. It's very nice. It's a intestinal area from the pig, from the swine. But you cannot eat raw because it has Yersinia. Yersinia is a big risk for that products, not really for milk. Listeria monocytogenes, it's basically is contaminated with ready-to-eat foods. We'll talk about later on. Vibrio parahemolytic, same as Vibrio chloride. Shellfish, jellyfish is a big risk. Clostridium perfringens difficile, we already talked about that. So, just want to let you know, this is the slides. We go there, we talk about is foodborne pathogen. This is the one. It is a little bit related to dental hygiene. Some of you will go to dental school, so this is a very good experiences for you. And this picture is really nice. Okay, you have a teeth. The reason will cause dental problem, they basically has two reasons. Number one is genetics. You can control that. Number two is dietary habits. If you eat rice all the time, or rice products all the time, then more or less, the sugar will stick onto your teeth enamel. When the sugar is stick onto the teeth enamel, if you fail to do the dental cleaning every day, or even the dental hand you go to see the dentist, what happened? They will feel the biofilm on the surface. Okay, our oral cavity is low oxygen to non-oxygen. Bacteria can use sugar. In the low oxygen environment, what will happen is fermentation. What the products of the fermentation generate is acid. And the acid could be demineralization of the animal, then will cause the tooth decay. So, very honestly to say, tooth decay is difficult to control because more or less the biofilm will be generated. And what the biofilm will be generated is Streptococcus mutans. And when you see the Streptococcus mutans, it's one group of the Streptococcus viridie. It's one of the examples. Okay, next one, chlorine. We just mentioned it very easily. Chloro gram-negative bacteria, common shaped, acquired from water-contaminated fecal sample. Shellfish is a natural reservoir. What's the problem? This is, we will talk about later on. We will mention this all the time. So, bacteria during the metabolic process, they simply coming from AMP, go to ADP, and go to ATP. This will generate an energy currency. For somehow, if this happens, generate CAMP. This looks like the money is locked in your wallet and you cannot use it. The frozen budget, they will all come from the problem is from CAMP. This is one of the examples. The chlorine toxin, the chlorotoxin, will activate adenine cyclase. So instead of AMP, go to ADP, ATP, they will have a cyclic CAMP. Then they block uptake of the sodium. And what happened? The osmotic balance of the lumen will be disrupted, and the lumen draws lots of the water. So you have a water diarrhea. You lose 10 liters of water in 3 hours. So chloral, if it happens, the rice water stool is a typical symptom. You need to do oral rehydration and use antibiotics immediately. And I want to tell you one more thing. why we have a whooping cough. The Bordetella hypothesis causes whooping cough because the cilia, they attracted with the sputum samples and they cause cough, and wound. The same is the cilium, the cell cannot go this way. They also activate cyclase, generate CAMP. Once the CAMP comes out, it will have a whooping cough. So it is the same thing. The principle is all come from there. And that is the simplest disease principle from the biochemistry level. We talk about that. Okay? So this is all we have for here. And we are not have a class on Thursday because we have a lab exam, so no lecture. And then following we talk about sexual transmitted disease.